So let's go ahead and go over this lecture quiz from today. See how we do. There are a few extra copies, but if anybody felt like they wanted a copy, but you could also always listen to the beginning of lecture, which even if you just wanted to just hear the first part, just to hear questions I thought were significant. Because you guys know Wednesday we had these multiple choice tests, and many of the multiple choice questions might come from this. So define histology is the study of tissues, and lists 4 primary tissue types or classes. They are epithelial, connective, muscle, and nerve. The tissue type that functions to line body organs and secrete is epithelial tissue. We also talk about it being avascular, and that it also is either simple, which is one layer lining, or it can be stratified. And you know the difference in the functions of those two. Simple is for rapid diffusion, and stratified is typically for protection, right? The tissue type that's involved in communication, receiving stimuli and then sending signals onto other cell types is what? Nerve. Distinguish between atrophy and necrosis. You said atrophy is the shrinkage of cells and losing some cells potentially, but necrosis is pathological cell death, and then tissue death, right? So that's a very different thing. Blood, bone, and adipose all belong to which tissue types? Connective, right? What type of muscle surrounds organs? It's under involuntary control, meaning you don't have to think about it, you can't really control it. And it controls organ volume. Would you say it is? Smooth muscle. That's exactly right. How do endocrine glands differ from exocrine? Endocrine secrete their products into the bloodstream and stay within. Exocrine glands secrete into the gut. Correct? Name the autoimmune disease that attacks desmosome connections between epithelial cells. That autoimmune disease is pemphigus vulgaris, pemphigus other types of it, but that's what it is. Name the pacemaker or cardiac muscle. It is the sinoatrial node, the SA node. Fair questions? Were they fair? Feel pretty good about those? If you didn't, then just make sure you get those before Wednesday. And then define metaplasia. Metaplasia means that a tissue has changed its function from its original function. It has changed, right? We typically think of that as not being a good thing, because it could indicate that precancerous type of condition, but in rare cases can happen. But these can, yes. So metaplasia is the term that means that. Were these fair questions? The lecture? All right. Okay. All right. Now, what I'd like you all to do quickly before we even just finish up the skin today is take a look at this picture that I gave out, these pictures that I gave out. And what I'd like to do is to do this. Actually, people who aren't here can maybe see this. Ah, look at that. Okay, that's better than my backside. Okay, see that picture. So anyway, let's do this together. Let's label the skin together. Should this be a really good quiz grade for people? It's sort of like that microscope quiz and the cell drawing. Those are kind of demi-lab quizzes, right? And you definitely want to do well on the ones that are like this, okay? So let's see if we can label the structures of the epidermis, which is actually, does everybody know where the epidermis is on this top picture? Does everybody know where the epidermis is on the top picture? It is this little part, right? This part that looks like they've got it kind of peeling up, don't they? That's the epidermis. Now, if we were to want to see those 5 layers of the epidermis, this is the picture we would like to look at. Is that correct? All right. Would anybody tell me, please, where the corneum is found in the epidermis? It's found where? The very top, the most superficial. So it's where this dead, like all of these, this is the corneum, isn't it? The most superficial. And then the lucidum is right under that. Lucidum. Then you have this layer that looks kind of dark and grainy. What do we call that layer? Granulosum. The granulosum. And then we have this kind of thick layer where the cells look like they're mature and they're doing their stuff. This is the spinosum. And then the bottom layer is called the what? The basale, which is the same thing as saying base layer. Y'all know that these layers, the term stratum is in front of it, but what does stratum simply mean? Layer, doesn't it? So if we go from the most superficial to the deepest, it's the corneum, the lucidum, the granulosum, the spinosum, and the basal or basale, right? Are we good with that? Any questions about the 5 layers of the epidermis? Let me ask you this. This isn't labeling. This is something you know just physiology-wise. Only one of these layers is found in thick skin. Which layer is that? The lucidum. And you all would know that for Wednesday, right? But you don't have to label this Wednesday, but I want you to label it now because we'll have it next, not Wednesday, Thursday. Excuse me. Was I saying Wednesday, Olga? I meant Thursday. That's what I meant. Okay. Y'all meet on Tuesday and Thursday. I've got to, okay, not Monday, Tuesday, Thursday. Okay. But anyway, you don't have to, this is not a lab quiz that you will have Thursday, but you will have it next Tuesday at the beginning of class. And you will just have to label what I am labeling with you today. Fair enough? Okay. All right. So we got the 5 layers of the epidermis. We know where to label the epidermis. It's this up here, right on this top tip. This top part is this is the epidermis. It's called the epidermis because it's sitting on top of the what? The dermis, which is this layer that has all the hair follicles, the sweat glands, right? So this whole region is the dermis, and that's sitting on top of the what? The hypodermis. So would y'all be able to label those 3 on this top picture? Would you be able to label the epidermis, the dermis, and the hypodermis? If you have any questions about them, now is the time. Alexis.
So for the 5 layers, are we doing the right side? Are you talking about the right side right here?
I couldn't care less what lines you use.
There's like 6 little
Yeah, I
dashes.
don't even care about that. It's the 5 layers. Make sure you know where those layers are. Some of the lines are pointing to cells. Look, let me tell you why I get a little bent about that, Alexis, and it's not at you. I promise it's not.
Well, I was
But
asking where did they go because there's 6 dashes.
yeah, okay. So they aren't dashes. These are layers. So you make your own. Okay, make your own. Here's what, let me tell you why I get a little bit bent about that. I've given lab quizzes before that are illustrations like this, and I've had students say, but I put them in the order that they were in on the left side and the right side. I had used a different picture with different dashes. So you know what those students had done? They had memorized the lines. Hey guys, these layers aren't lines. They're layers. Look at a lot of other pictures, then look at this, and you make your own thing. And it better be something I can kind of see too. Because are you going to necessarily get this exact picture? No. By the way, no. So don't worry about the dashes and the lines on here. Make sure you know where the layers are and what they look like, and look at several different pictures. Let me tell you about people. What'd you say, Alexis?
So that's why I asked because I didn't know.
Okay, yeah, and I appreciate you asking, because I really, truly do appreciate you asking, because this is where students make mistakes. They make mistakes in thinking they can memorize a picture like what's written down the left and what's written down the right. That's not a structure. A structure has shape and definition, and layers have certain appearances. Don't memorize lists. Don't do it. Look at structures and know no matter what picture you saw or what structure. If I were to show you all epidermis histology slides on people, if I did it myself and Aaron and Nicole, would the 3 of us be exactly alike? Would our histology slides look exactly alike? Would they have dashes? Would they have dashes in those layers? No. So y'all have to get rid of that mindset that I consider K through 12. Get rid of that. You all are look, And I appreciate the comment being made because I really did. I didn't realize people did that until I had some students come up to me and say that very same thing. And after the third or fourth one did it, I realized that students were really doing that. When they were learning anatomy, they were memorizing lists and not looking names to structures. Do you get what I mean? So please, I'm giving you this as a just one example, but you should be looking at a lot of examples. This is an illustration, but go on and look at the true histology slide, right, that you have in your textbooks. Google them and see, because you don't know which pictures I'm going to give you. And I'm going to try to find something that doesn't have any lines for that very reason. For that very reason. Okay. So, like the top picture doesn't have lines, which is great. Okay. So we have those three, the epidermis, the dermis, and the hypodermis. That's great. Would everybody know when we look at this hair, we can see a couple of them, but when we look at them, do we know what it's called when it's above the skin? What's that called? A hair shaft. But below the skin, it looks like it has, like it's encapsulated almost. And what is that called? The hair follicle, isn't it? On this hair follicle where you see a coronal section, now there's one that hasn't been cut that way, so you don't see the coronal section here. You sort of see the whole thing intact. But the one that has the coronal section, it looks like there's almost like a little flask attached to that hair follicle. What is that gland called? It's a sebaceous gland. If you look at this sebaceous gland intact, you can see it here intact, but here it's been cut, hasn't it? So we look at a lot of different ones. We're not just, we look at a lot of different ones. And we see the sebaceous glands all kind of have that appearance, don't they? All right, so that's sebaceous gland. Then we see this, it almost looks like a little coiled duct that is entering into the hair follicle as well. But we see one that looks kind of similar to that, that's not going into a hair follicle, that's going straight to the surface, right? But what are those glands? What are those glands? They're sweat glands. Those are sweat glands. I don't care that you know apocrine versus merocrine, but they are sweat glands. And they look very different than sebaceous glands, don't they? Do they? And then there was something else that we learned called the Pilo Erector muscles. What are the Pilo Erector muscles? Well, they aren't goosebumps, they're muscles. But if they're contracted, they give this sort of appearance that we call goosebumps, but they are little muscles. And if you look at this, even this illustration kind of looks like a little muscle. And is it attached to the hair follicle? Is it? That's the Pilo Erector muscle. That's exactly right. And then you see pores. You see pores that are visible from the corneum. The corneum, which is the most superficial layer of that epidermis, you can see pores, can't you? All right, guys, that's all you have to label. Now, would you want to just look at these two? No? You want to look at a lot of different pictures. You want to know that it won't matter where the lines are, as long. And let me tell you another thing. I really, what? But anyway, let me say this. Please keep this going forward in your study of anatomy. Just because you see a picture with a line going to that little one site doesn't mean that it's necessarily just that site. It can be the whole area and region. That's why you should look at tons of pictures, right? And tons of aspects. You should look at anterior, lateral, inferior, superior, right? You should look at a ton of different aspects and make sure you understand. Just because you saw a picture where a line was going to one spot, that might be messing you up, right? All right. So we have 5 layers of the epidermis. We know we have 2 layers of the skin plus what they're sitting on. The hypodermis they're sitting on, but we know the epidermis and the dermis. We know hair follicle, sebaceous glands, sweat glands, Pilo Erector muscles, hair shaft and pores. Is that right? That should be, is that good? All right. So look at, like I said, look at quite a few different pictures, because your lab quiz where you are labeling those structures might not be these exact pictures, but that would be all right. Right? All right. All right. So, great. All right. Now today, we're going to finish up the skin. I had hoped that you guys, by today, are feeling really confident about genetics and also histology, because those multiple choice questions will be found on the test Thursday. And then the skin, we've talked about a lot of things too. Do you think you'll have questions about like the markings of skin? Yes. Yep, I think so too. I actually don't know how far we got in this. How far did we get?
To sweat glands.
To sweat glands? Okay, so we hadn't talked about hair or anything like that? Or had we? Oh, we had talked about hair. And so we were way down. Okay, I see. So were we like right here, you mean? So kind of right here. And we had talked about different kind of glands. So we've got the sweat glands. The apocrine glands don't come until like puberty. We've got sebaceous, which means sebum, which means oil. Ceruminous, make sure you know, earwax, right? And do we know the functions? I think we had stopped on that to talk about functions too of earwax. It's a repellent. It waterproofs the ear canal. It also keeps the eardrum flexible. Mammary glands, what do they secrete? Milk. And then we had talked about these sweat glands. This is great. And again, here's some slides on each of these types of glands. When we think about mammary glands, we do know that, that bothering you? Okay. When we think about mammary glands, we know that typically, you know, in most mammals, we have these two rows. Primates usually have only anteriormost glands, so two. But some people can have additional nipples. Do y'all know? It's actually not uncommon. Do y'all know anybody with this? This is called polyphilia. Do y'all know anybody with this? No? I do. Yeah. Okay. So it's actually pretty common. And sometimes people think it's like mole or some erect, but then they realize it's just an extra nipple, and it's not a big deal. And honestly, the ones I've seen have usually been in males. Is yours male that you know? Female? Okay.
I know a guy that had a little down here.
Yeah. So it's likely, it could be slightly to the, yeah. So it's not a big deal, but we think about it too. So look, I want to talk about this idea that skin is our largest organ, isn't it? It's our largest organ, and it is an organ that is creating this external barrier to our environment. And so much can be absorbed through the skin. It's actually kind of crazy when you look at the percentage of what we absorb through our skin. So we have to be aware of that, right? That we can do that, but it's also a way that we can it's excretory, so we can get rid of toxins through our skin as well. But maybe it makes sense to you that since it's our external barrier, it's the most vulnerable to injury. So as we age, does it take a beating as we age? Yeah, kind of it does. And so anyway, we kind of get that. So we know that skin diseases as we age are really prevalent. Now, they're not all created equal, obviously, not all skin diseases, and sometimes there's hereditary components as far as some of these skin diseases go. But when we think about cancer, let's keep in mind that not all skin cancer is equal. I've had people who I know who had like squamous and basal cell cancers, and sometimes these cancers that you think of, they can get removed. How many of you know people who have, like twice a year, they'll go and have these types of cancers removed? And they go and they get, several removed a year. Now, which is a really good thing that they do. These squamous and basal cell cancers usually are going to have an appearance that is going to cause somebody to be alerted that they're there. They're going to be like a sore that won't heal, or they're going to be something that might be a little bit itchy or oozy. So usually if something is happening like that, does it get our attention? It gets your attention, and because it's not healing, you go see about it and you get it removed. And I kind of not that I want to tell the people that, you know, have had these types of skin cancers that it's no big deal, but kind of no big deal as long as you go get it done, right? And stay up with it, because that would be a really good thing. Now, on occasion, these can also metastasize. And where did we say cancer deaths come from? Does it come from cancer when it stays in its local region, or does deaths come from what? When it moves from the original location, when it metastasizes. So these two, we don't tend to think about that, because usually you can see they're going to cause things that make you go see about them. They're slower growing. They typically are associated with overexposure and beaten down skin, you know, from the sun. And you're just going to see about them, and they're going to be slower and metastasizing. The one that's deadly of all ages, not just older people who've abused their skin because of occupation, and I don't mean intentionally abused, but, you know, occupations, farmers, people who are out in the sun a lot, and whatever. The one that's at all ages, it really, teenage to all ages, and it's deadly, is malignant melanoma. This is the one you just don't want to hear about. And by the way, this originates in the melanocytes, and this can originate where the sun doesn't shine, if you know what I mean. These can be under your scalp, so under your scalp. It can be on your scalp, under your hair, under your hair, under your scalp. So you don't see them. Or it could be like in pubic hair, or in creases and folds that you don't see. These can be on your soles of your feet. And actually, those are really scary, the dangerous ones. Under nails, toenails, fingernails. The reason I hate some of these pictures is because sometimes you see these pictures and you think this is what it has to look like to be melanoma. But it doesn't have to be this glaring, obvious look like this. But, you know, you do hear about the darker the mole, the more worrisome. But there can be melanomas that are light gray. Light. So let me tell you about, and this is already old information. Let me tell you about the ABCDE rule. And E is in there too. ABCDE rule of your moles. Did we say that everybody has a mole somewhere, right? So your moles, you should map your moles, and you should know your moles, and you should frequently visit your moles. Because what you want to do is this ABCDE thing, check on all your moles. And they say twice a year is a good thing to do with your moles. The A stands for asymmetry, meaning that if we cut a line down the middle part, you know, whatever, a line that bisects it, then they're not symmetrical. If we folded it, it wouldn't be symmetrical, right? B stands for border irregularity. And if the border is irregular, it's not going to be symmetrical. But do you see how this border is irregular? C stands for color variations. So it doesn't necessarily mean just black, dark. It can mean that there's there's differences in the color dispersing. Do you see how there's lighter areas in this? So the color is varied within it. And sometimes it's just like the color's washed out within it. But anyway, color. D stands for diameter, meaning that it shouldn't be bigger than 6 millimeters, which is the size of a pencil eraser, a normal pencil eraser. So if the diameter is greater than 6 millimeters. And E is probably the most important one. You ready for it? E stands for that it's evolving, which means what? It's just changing. It might not be growing. It could be that the color is changing. It might not be growing. It's just that it's changing in some way. So you've got the ABCDE rule of your moles. If 2 or more of those letters occur for your moles, you're supposed to get them checked by a professional. So you don't need all ABCDE, do you? You just need 2 or more of those things, and your moles should be evaluated by a professional. Fair enough? This happens in any age. It can happen in teenage. it's rapidly progressive. It can happen any age. So it's not just older people who have, you know, the skin's been kind of abused. Please know that. Any questions about malignant melanoma? It has a really high, grows fast, and it has a really high mortality rate. The moles themselves don't grow, it's just the cancers grows fast.
Well, I have a couple of patients, older patients. One day I'll be taking care of them, and that pops up. And
Usually that's not melanoma. That's not the melanoma. Usually it's one of the other ones that do that kind of like that. Melanomas are usually kind of, they don't just, do you know what I'm saying? They're not going to be like these raised, oozy, they're not going to be like that. So I don't know which patients have exactly, do they call it melanoma? I can tell you that a lot of, if somebody has those squamous and basal cells, they're getting them removed, they're getting them done, and they put a little patch on them, and then they check the skin every 6 months or so, and they remove more, you know. Melanoma, if you have melanoma, you're getting a margin taken, you're getting that removed, but plus a margin, plus lymph nodes, and every 3 months you're getting complete body scans to see if the cancer is metastasized to your brain, to your liver, to your bones. I've had a lot of people who say they have skin cancer, and you know they say that it was melanoma, but it's not. You'll know it's melanoma when somebody's having complete scans every 3 months for like the first few years. And if they're negative, then they can go like once a year, and then after 5 years, I think they go, or twice a year, and then after 5 years, they go like once a year. It's a serious.
So is that when they take like or not margin, they take a chunk out of them?
Yeah,
they have to get up under and
yeah, they have to get a margin all around and underneath, and then sometimes they also try to see about lymph nodes, depending on where it is. Yeah. So it's not like the squamous and basal types. So now, what causes the skin cancers we do think are UVA and UVB rays. So UVA and UVB rays. So the answer to that is to try to protect skin, you know, from direct exposure to the sun. You don't need direct exposure, right? Our melanocytes do a really good job typically. So if somebody's out in the sun long enough, they're starting to tan, aren't they? Because that melanin is trying to protect, but it can't completely protect. Now, obviously, lighter toned people are more at risk for those cancers than darker toned people. But darker toned people have had melanoma too. They have. All right, so burns are very. So anyway, but different causes. You know, y'all know this. Y'all know different causes. You know, certainly hot water or liquid, sunlight, radiation, electric shock, acids, bases. So so any of those things can. Now, the cause of death. This is what I want you all to hear me say, because a lot of people think, oh, the risk of having severe burns, meaning that you have a certain percent of your skin loss, is going to be infection. You might not live long enough to get infection. The initial concern with burns is fluid loss. Please know that. Now, there's something with burns called the rule of nines, and in the rule of nines, they have designated certain regions of your body as a part, if you will, into 9 parts. Now, if you have 2 of those 9 parts that are done, you're said to have a 20+% burn. If 3 of those parts are involved, then it's the 30+%. Y'all have all heard that so-and-so had a 40% burn, you know, whatever. So that means it was significantly over the body. So if you've lost that much of your barrier, you're weeping fluids. And you might not see them. It might not like be dripping, but you are weeping fluids. Fluids are you're losing fluids. So it's fluid loss. That's the initial thing that's going to kill a significant burn on a patient. It's going to be fluid loss because that fluid ultimately comes from your blood plasma. And blood plasma, if the blood plasma is losing volume, your blood pressure is doing what? Dropping. This leads to hypovolemic, that's with the V, hypovolemic shock. Hypovolemic shock. Hypo means low. Volume, bowl. Emic as a suffix always means in the bloodstream. Low volume in the bloodstream. It's the most common shock. And I want you all to know that shock means, medical shock means dangerously low blood pressure. That's what it means. Y'all have all heard of somebody going into shock, haven't you? Mm-hmm. What that means is their blood pressure is dropping so fast that their nervous system can't compensate for it. And if the blood pressure drops like that, is blood being impaired and not flowing to the tissues? Not flowing to the tissues. Is the heart going to try to pump faster? The heart's going to try to pump faster. But the heart will also at one point get a signal to just do what? Stop. The danger of shock, medical shock, is cardiac arrest. For that very reason. Does everybody understand that sequence? So medical shock is a serious thing. It means that blood pressure, for whatever reasons, have dropped. And if the blood pressure has dropped, the heart might get a signal to just do what? Stop. This is why when you see somebody's blood pressure dropping like that, you've got to be ready to restart the heart. Don't you? Okay, but you also got to be ready to bring that blood volume back up. Right? So burns, the most initial danger in burns is fluid loss, equally hypovolemia, equally shock, equally risk of what? Cardiac arrest. Does that make sense? Now, if you can live through that, if you can get somebody who's had a burn, significant burn, and you're replacing fluids, which is going to be tricky because they're losing fluids. And you've got to be replacing those fluids. If you can get those fluids replaced and you've got them back in homeostasis as far as their blood volume, great. Now what do you have to worry about? Infection. Fair enough? Mm-hmm. All right. So that's really, really true. How they are judged is by what sections of the body are involved and how widespread that might be. But they're also you look at the degree of depth, right? So you think about partial thickness versus full thickness, we think about, and y'all have heard of, and you'll hear it referred to as partial versus full thickness, but you'll also hear first, second, and third degree. First means it is into the epidermis, and it's going to be kind of painful, kind of red, maybe a little swelling. But if it's gone down into part of the dermis, it means it's blistering. There's a blister, like that one. Right? I can't complain about that. That's just a little bitty thing. But okay. So, but it's blistering, right? So if it is third degree though, this is called full thickness, and you have gone down to the hypodermis and you have lost a huge margin here. This is definitely going to require it's going to have scarring, fibrosis, which we know means scarring, don't we? Mm-hmm. And it, depending on its margin, how big it is, we might have to do grafting. So this is serious, third degree. So fluid replacements first, and then you worry about infections afterwards. And again, this partial thickness versus full thickness. I would maybe say that this guy how bad this is. This might end up blistering tomorrow, right? So, yeah. But anyway, so this is kind of what this looks like, and it's just an idea. UVA and UVB rays both can lead to cancers. We know. I put this in here because it can be misleading. It says, as the sales of sunscreens has risen, so has skin cancer. What would that make you think?
Sunscreens are
Sunscreens are what?
the reason.
Yeah, so that really kind of makes you think, well, are sunscreens causing it? Now look, sunscreens can have things in them that are carcinogens, but we no longer really use PABA. Y'all have all seen on those sunscreens PABA free or oxide free or whatever it'll say, but there are usually still some things in there that if you were to bathe in these things at huge quantities 24/7, they could lead to some issues. I don't think there's a single dermatologist you could probably find that would say not to use sunscreens. They're saying we need to be using sunscreens. It has a lot to do probably too with the thinning of the ozone, right? And other factors where people now, instead of people going out in parts of the day that's not so hot, we have people now that are kind of worshipers, sun worshipers, right? And are intentionally tanning and whatever their skin and stuff, where, back in the day, people didn't do that. They went out early, they worked late, they had siestas in the mid parts of the day. They didn't go out when it was too hot or whatever for a lot of different reasons. But now people have kind of changed what they do, so we're seeing more and more of it. I don't ask you about these terms, but you should know these prefixes. Auto means self, doesn't it? So autograft means that you're getting it from a different region on a patient. I always thought that was so insulting. I really did. Like if you've ever gone to a burn unit, a hospital that has a burn unit and seen some of those burn patients, it's really hard. It's really hard to see what they go through. It's a lot. They go through a whole lot. And sometimes you think to yourself, they're going through so much with their injury, and then you're going to insult a healthy part of themselves. But I can see why you want to do it, because it's less chance of rejecting the graft, right? So it's a hard decision to make. ISO means you have an identical twin. Do many of us have that we can go to? No. Now we can culture skin patches now, but is that going to take some time? Yeah, so that's going to take a little bit of time. And then there's these other, homo means, homo or allograft is grafting from an unrelated person, but another person. Hetero means different, doesn't it? Or sometimes that's referred to as xeno, which is going to be from another species. So you hear about pig skin and other skins that you can get from another species. But anyway, so these are just some different types of grafts. How do y'all, and I don't think I asked you about those anyway, but would you know the prefixes? Yeah. Okay, good. All right, so yes.
Do the temporary grafts, like when they make that option to make a temporary one or put one on one, does that act like a Band-Aid? And
kind of, yeah.
Pull it off.
Yeah, they act like that's exactly what they do. It's just temporary until they can get some keratinocyte patches done, or until the person's skin has gotten a larger margin. Sometimes you'll see. Have y'all ever seen when somebody's had some of this? They'll open up the peritoneal cavity, they'll keep it open, and the person can put their appendage in. I mean, if it's usually the arms or hands, and they can put it in there. They do all kinds of stuff, and I don't know all of the reasoning behind it or whatever, but they've obviously had some positive results if they do that. Yeah. So anyway, your skin, we take it for granted, don't we? We understand, and I do want to just bring this up. I know y'all already know it, especially when people have done a lot of CNA work in those types of facilities. You guys know a lot of medications are delivered through the skin in patches. So medications can be given that way, which is a great thing. You have to be really, really careful. Have people had overdoses because they thought they pulled off a patch, but they hadn't, and they still had patches on, and then they put on another patch and realized the other patch just slipped, and all of a sudden they're exhibiting signs of overdose. Sometimes they don't make it. Yeah, so they have. And also we get rid of toxins. How many of you had, maybe not patients, but just friends that you can smell toxins they're losing through their skin, whether it's alcohol or nicotine? That nicotine could be from external, but it also could be from internal. How many of you you've seen that? You know that it can be like that. So it's an excretory organ. It's an excretory organ, and it's a great organ for delivery of meds. But people have to have caution with that. So what we put on our skin, we got to also be thinking about that it can be, a lot of that stuff can be absorbed. How many of you love bubble baths and lotions and facials? Facial, it's probably pretty obvious. I haven't, but also like makeups and stuff, which I use makeup. Y'all are probably lucky that I use makeup. You wouldn't want to see it. So anyway, but no, should we be thinking about, like, are those companies vetted? Are they being careful what they're putting in their lotions and stuff? I guess they kind of are, but you know, probably not most of us really question it too much, do we? So anyway, all right. So we're good? Y'all feel good? We have genetics, histology, and the skin. So this would be a good time for you guys to look back. Let's take chapter 4 for a second. Before our little break this morning, before the break that we're going to have in about 15 minutes, let's think about each of those chapters. In genetics and inheritance, do we know the 2 steps that get us from DNA to proteins? What are they? What are they? You say it like you own it. Transcription and translation. Do we know where they occur and what the outcomes of those are? Do we know that DNA molecule and the 4 bases, nitrogenous bases that make up that molecule? And we know that DNA is double-stranded with billions of nucleotides, which is a sugar, a phosphate, one or more phosphate groups, and a base. And there's just a ton of those, 2 strands, right? Do we know the law of complementary base pairing? What does it say? Adenine will always be base paired with thymine, unless you're talking about RNA, which is within the uracil, right? So, yeah, guys, these are going to help you answer some questions Thursday, not Thursday. So great. So DNA, you know the molecule. Do you know who's given credit for the identifying the structure of the molecule? Watson and Crick and Franklin, really, but Watson and Crick were the 2 who really got the credit for it. Franklin died. So anyway, John, I didn't know it was John. So great. And then we said the structure allowed for us to then just blow up as far as how much we knew, because that's what helped us to figure out transcription and translation, and understanding that 3 bases code for a particular amino acid, and knowing all 3, knowing all the combinations of 3 that can happen is called a what? It's called a genetic code, isn't it? Knowing all of the codons. How many codons are there? 64. That's called the genetic code. Knowing each sequence, which amino acid it calls up. How many different amino acids are there? 20. There are 20 different ones. That means that some sequences call the same amino acid up. But what we know is that when a mutation occurs on the DNA sequence, meaning those 4 bases, when the 4 bases when there might be a deletion of a base, or an insertion of a base, or a switching of a base, that may potentially change the codon. If the codon changes, but it didn't change the amino acid, that's just a benign mutation. But if it does change which amino acid comes up and gets in line, could that be deadly? Or could it be beneficial? Could it? Or it could be beneficial. Or it could be just that it's going to make a variation in the trait. Right? So we know not all mutations are bad, but we tend to think about them being bad because they're the ones we think about. Okay. Okay. If I say something that's not making sense, stop me. This is a little bit of a review about chapter 4. So we understand getting from DNA to proteins. We understand we inherit our DNA from our mother and our father. We understand that we have 23 pairs of chromosomes. And those pairs are called, when you think about a pair, they're referred to as homologous partners, aren't they? So we have these homologs in each of the pairs. We did karyotyping. You remember that? Karyotyping is physically looking at the chromosomes And there are dozens of anomalies or disorders that you can diagnose from just looking at the chromosomes. Does it tell you what's actually on the chromosome? No, no. But there are dozens of disorders you can figure out from karyotyping. Now, those traits that we think about, because we inherit one half of our DNA from one parent and the other half from the other parent, those traits that are on the first through the 22nd pairs, we know we're going to have at least 2 chances at the trait. Now, if it's a trait that's influenced by many different sites, many different other pairs, then we know that that has a lot of alleles contributing, and that what we usually see about that trait is we've got a rainbow kind of effect in the population, right? But some diseases aren't. They have very specific locations, loci. And honestly, you know, there's many traits that are like that, that are inherited that way. But if it's the first through the 22nd pair, the autosomes, then you know you're going to have at least 2 chances at it. If for a trait you inherit the same information from each parent, what did we call that? Being what for that trait? We called it being homozygous for that trait. You might be homozygous dominant, or you might be homozygous what? Recessive. That's exactly right. If you inherited different information about that trait from each of your parents, and we call that what kind of condition? A heterozygous condition. Is that right? All right. So we said there's a way, and you know, we use this for disorders. We don't really use this for cuteness. You know, we use this for disorders. But there's a way that if you know the parents' genotypes, you can figure out the probability for that trait in each of the matings. Each time those same parents mated, you could figure out the probability of their offspring's genotypes. What tool was that? It's a Punnett square, right? And you all will have a couple of questions that you will use, and you'll write on your test. Just write your genotypes of your parents, meaning you knew both what was on both homologs, right? Write your genotypes, and I'll be giving you that information if you know how to read the question. I'll give you that information. And then you'll have to figure out if the mom is this genotype, you know she can give one or the other to her eggs, right? And so you put the eggs on the top, you put the sperm on the side, and then you have a little box with 4 squares. What do each of those squares represent? A possibility of a child. It's a child's genotype. Each of those squares represent a child, potential child, right? So then you can answer questions about the probability. So, right, who's the father of genetics? And right, so anyway, we know the difference between genotype and phenotype. Phenotype is just what you see. So somebody's either dominant for a trait or recessive for a trait. Which phenotype, listen to this question, which phenotype gives you the genotype? Recessive. Because if somebody's phenotypically recessive, could they be holding a dominant set of information? No. So that gives you their genotype, right? If somebody's phenotypically dominant, though, they may be genotypically heterozygous, or they may be genotypically homozygous dominant. You don't know that. You don't know which one from the phenotype that they're dominant. Okay, so that's just about some inheritance things. We talked about traits blending, because they do that, right? No. They do not do that. That would be like saying your DNA blended. It doesn't. Your DNA is a molecule. And those traits, those recipes, those sense strands are not blending. They're holding true. Now, might traits be hidden for a few generations and then come back out, or, you know, situations happen that allow them to be seen later? Yes, but they're true. They do not blend. Right. All right. So we talked about codominance, meaning referring to traits that if you have information that's different from each parent, you're still going to express both of it, right? And the best example of that is ABO blood typing we talked about. Yeah. right, so that was our little chapter 4. How are y'all feeling about chapter 4? Did that seem like a long time ago?
Yeah.
Okay, it was like last week. No, it wasn't long ago, but okay. Then we had histology, 4 tissue types. We said the DNA dictates what happens in the cells, and as cells differentiate, that means they're maturing and they're going to do certain functions. And so we looked at those cells' functions, and we said that cells, if they function in a particular way, cells make up tissues. We've got these 4 classes of tissues that they make up. And the reason we said we want to know this is because there's certain disorders, autoimmune, infectious, inherited, that attack specific tissue types. So we wanted to know these 4 tissue types and what their special functions are. Epithelial, we use 2 words to describe them. If you ever see these 2 words, you're going to know it's epithelial. What were those 2 words? Line and secrete. Muscle, we said 3 types: cardiac muscle, skeletal muscle, smooth muscle. We said that they're unique because the entire cells that make up fibers can do what? Contract, right? They can contract. And then nerve, we said, are so special because nerve, 2 types of cells in the nervous system. What were the 2 cells? Neurons, they get all the glory because they detect and they send signals. And they're those huge, octopus-looking cells. But what other cells? Which are the what to the neurons? The support or helper cells to the neurons, right? And neurons detect signals. We talked about homeostasis, and we said you have these set points, and you have a way to detect when something's getting high or getting low. Well, what was the way to detect? Neurons that are specialized, that are detecting when things get high or low. They detect environmental stimuli, and then they respond by sending signals to other neurons or endocrine glands, which are epithelial, or muscle, which can contract. But neurons are kind of cool because they're our great communicators, right? And then we had the most abundant tissue type, which was connective. And it's the most diverse tissue type. It's the most diverse. When we think about connective tissues, there's certain cells we think about, like red cells and white blood cells, because blood is a liquid connective tissue. We talked about ligaments and tendons, didn't we? We talked about elastic and fibers and collagen, didn't we? We talked about bone and cartilage and fat. Is that a good thing or a bad thing? Let's think about it as being a good thing right now. Okay, yeah, obviously. We said anything of the good things we can have too much of. But yeah, okay. So it's a good thing, though. It's a storage molecule for energy. It's a storage tissue for energy. It insulates. It stabilizes organs. So a good thing we think about. So adipose, cartilage. By the way, you need to write this down because I don't think I gave it to you. We want to add it to those notes. I'm so glad I thought about it. Cartilage. Again, does cartilage help to stabilize structures? Does it? Think about this, does it? Mm-hmm. It really does. There's cartilage found between our bones that cushions, stabilizes, gives structure. Cartilage is made of cells called chondrocytes, C-H-O-N-D-R-O, chondro, C-H-O-N-D-R-O, cytes, C-Y-T-E-S, chondrocytes. So what would chondroblasts be? Early chondrocytes. That's what they are. So they're the early chondrocytes. So good. So good job. What would chondritis be? Inflammation of cartilage. Right, cartilage. I don't know if the costal region was in your regions or not, but costal means ribs. So if you had costal chondritis, what do you have? Inflammation of the cartilage is holding your ribs to the sternum. So do you think people are in pain when they have that costal chondritis? Yeah. So learning those prefixes, learning suffixes, root words, is that all really good? makes things easier after you get those down. Right? Like while you're getting them, it might seem overwhelming, but as soon as you start getting that language, good. So great. Cartilage is one of those rare connective tissues that is actually also avascular. And we usually think about avascular as being associated with epithelial tissue, but cartilage is avascular too. And so this is why injury to cartilage can take so very long to heal. Really long to heal, right? Speaking of healing, we talked about regeneration versus fibrosis. Regeneration meant that what was going to happen? You get your function back. Regeneration. Can all tissues regenerate? No, it's depending on the tissue type, right? Some tissues really don't have that ability. Others pretty well can do that. But it also depends on the magnitude of the injury. So if fibrosis occurs, we said that's another way of saying what? Scarring. So fibrotic events mean scarring. That's another. Fibrosis is a process of scarring. And when that happens, it means that you're going to really mostly get these connective tissues that come in and replace and lay down and replace what tissue had been there. You're not going to get it back. We talked about keloiding. What was keloiding? Great. And then, and now we've been talking about skin. That is our first organ. Now, what I'd like is if, before this quick break, the break won't be quick, but it'll only take a minute. But before we go on break, you guys, the disorders table that's at the end of that chapter, the disorders table that's at the end of that chapter. There's disorders listed in there, and you wouldn't want to, did you copy it there? I'm sorry. Okay, well, actually, all right, thank you, because I can't see that. That's so little. Can you read it? Okay, all right, I can read it too. I get up.
Yes.
Okay, so I can see it. All right, so, sorry, I took yours, but, so great. So we look at this disorders table and look at acne. Acne is actually something that we think about at puberty and after, right? So you don't ever think about acne before puberty, do you? But it's usually because of those hormones at puberty are affecting the sebaceous glands, and they're releasing a lot of sebum, oil, and this is changing these tissues enough that it can set you up for bacterial infections. It's actually a bacteria that causes this. I don't know if they give it to you. They don't, but anyway, it's called Propionibacterium acnes. It's a bacteria, and it can cause acne. Now, can acne cause scarring? Mm-hmm. It actually can. So cystic acne can actually lead to scarring. So this can be, it can be serious. And then eczema and psoriasis. I told you the difference between eczema and psoriasis. Dermatitis, this is actually just can be a lot of different things, but it's an inflammation. So inflammation of the dermis, and it can be for a lot of different reasons. I had a student actually she's not a student of mine this time, but in the fall who was having reactions to gloves where she was working. And those of you in healthcare, you've got to be able to wear gloves, right? She had about the worst case I've ever seen. And you guys y'all don't know what I would never say who it is, but she wouldn't mind coming in here and showing you though, but I'm not going to say who it is. But I don't think she's going to be able to work. It's about the worst reaction I have ever seen. And so far, they can't find any gloves she's not responding to. Her hands are double the size they're supposed to be. Imagine your hands double the size. They're so cracked and so irritated. I don't know how she's going to do this. But anyway, eczema and psoriasis. We said eczema is a hypersensitivity. Psoriasis is an autoimmune. Those are very different, distinctive causes. So please note those types of things, whether something is autoimmune versus hypersensitivity versus infectious. What does autoimmune mean again? Just to be clear that everybody knows. It means your own immune system is attacking your own cells. That's exactly right. Seborrhea, that y'all can look at that you can see sometimes with the dermatitis. Tinea. I want you all to know if you hear that somebody has a tinea infection, it means that they have a fungal infection. So there's tinea versicolor, which can cause like just, if you ever see somebody tanning, sometimes they'll look, or darkening, they'll look like they have these almost like little circular lighter patches on their skin, and it's because the fungus on their skin is kind of taking over because the skin's been damaged. Tinea capitis means like dandruff. Tinea pedis is athlete's foot, right? So these are types of things. Are they listed in there? Oh, okay, but anyway, so tinea, you just hear about tinea, it means that there's a fungal infection, which is usually pretty easy to treat. You give an antifungal cream, right? Ringworm. Ringworm is a fungal infection. Has everybody seen ringworm? So that is actually one of the reasons it's called tinea. Fungal infections are called tinea because there's an actual worm that is named that. Okay. So we talked about alopecia, didn't we? We talked about birthmarks. Alopecia is what? Alopecia is losing your balding, right? So we talked about birthmarks. We said hemangiomas and also port wines. We talked about hematomas, did we? Genital warts and any wart. I don't know why it just says genital. It should say any warts. Please let's write it down. Warts are caused from human papillomaviruses, HPV. HPV. Human papilloma viruses causes warts. So anybody who's ever seen a wart or had a wart, it was viral. It was a virus that did that to the skin. Most warts are going to be self-limiting. Most are going to be gone within a couple of years, you know, but some people can be really prone to these types of skin viral infections. Some HPVs only attack the hands. Some HPV viruses only will give you warts on your feet. You know what they're called? Plantar warts. Plantar warts. Some HPVs will be, your genitalia and oral mucosas are very much at risk. There are more than a dozen plus that cause those types of situations. Most people who are HPV positive have never had or seen a wart. You do not have to have had a wart or seen a wart to be positive for this. So the only way to know is to be checked, right? So the thing about genital warts is that there are a few of those dozen or more that can lead to mutational changes that lead to cancer. They can lead to cervical cancer, oral, throat cancer, neck cancer, so they can lead to cancers. So that's why all people, especially middle school, starting at middle school, are recommended to have immunizations against them. So there are immunizations now to protect people from some of the serotypes of human papillomaviruses that cause warts. I worked with a young girl. I was the same age. Okay, well, anyway, because when I remember her, she seems young to me now, but guess what? She looks as old as I do now, wherever she is. I haven't seen her in a long time. Actually, I saw her about 10 years ago, I guess. But anyway, I worked with her when I was first out at college, and she worked in pathology. And she said she had it since puberty. Her hands look like you know, you dip your hands in wax maybe, and you got like, you know, you dip your hands in wax. She looked like she dipped her hands in wart juice or something, because her hands, and it stopped right at the wrist, both hands, it stopped right at the wrist. Beautiful girl, this girl's a beautiful girl, but right at the wrist, her hand, and she had hundreds of little tiny, not very raised warts. Not all warts are created equal. Y'all might be thinking of warts like on that show, what was her name, Nanny McPhee or whatever. No, I'm not thinking about those. Like these were like you have to, at first when you looked at her, you thought, it's on her hands. And then you realized it was like hundreds of warts on each one. And they stopped right at the wrist. I've never seen it before, and I've never seen it since. But she said since puberty she had that condition. So some There are more than 100 different serotypes of HPV. There's a lot of different types of viruses that can cause warts, and some of them are really isolated to certain body parts. So, okay, human papillomaviruses. The thing we worry about are the ones that can lead to cancerous changes. Okay, so y'all look at even things I did mention. Look at those and make sure you're kind of familiar with them. So you look at the term hirsutism, hirsutism, excessive hairiness, right? Excessive hairiness, right? All right. Y'all feel like we can take a little break and then we're going to come back. All right. I do want people, if they want to, at 11, I want you all, or at 11 or so, no, maybe it's not 11. Yeah, it is. It's going to be 11. We're going to maybe go over to the lab. We can't get in there until about 11:05. And y'all can look at those slides again, because on Thursday, are you going to have to be able to see a field of view and tell me which tissue type you're looking at? Will you get full credit if you just say which of the 4 tissue types it is? Yes. Full credit. Today, if y'all want me to, I will go through some slides and give you classic fields of view and let you all look at them. Okay? Yeah. That would be lovely. Somebody said, okay, now let me say this. After this 15-minute break we're going to take, I am going to pause the recording. I actually might stop it and just start the next one. I am going, when we come back in here for a little bit, we're going to talk about bones. We're going to move on. But let me say this. Are you going to have anything about bones on the test Thursday? No. No. All right, take a 15-minute break and come back. Don't forget to come back.